Darwinian evolution needs time. It needs lots of time in which to work. Time that in 1900 didn't exist, according to the physicists, because the Earth and the Sun were simply not old enough based on the knowledge of that time. But as people began to understand the phenomenon of radioactivity, then all of a sudden time came back. Time for Darwinian evolution. But in order for Darwinism to become the dominant paradigm that it has become, we also indicated that it needed a theory of inheritance, a discrete theory of inheritance that could allow spontaneous variations to accumulate in a population and not be swamped. So first, let's note that genetics, as we understand it, is just one type of a theory of inheritance. There are other types of theory of inheritance. So theory of inheritance is not synonymous with genetics. Genetics is just one of them. And what we mean by genetics, thanks to William Bateson in 1903, is the name for Mendelism, Mendelian genetics. The monk, Austrian monk Gregor Mendel, who in 1865 published the results of the experiments that he had been doing with raising peas and crossbreeding and looking at the way color and skin texture changed as a function of generations, looking at the patterns that he got, came to the conclusion that there was something in the plant, that was something in the seed that contained the determinant of the properties like skin color, color and skin texture, whether they were smooth or wrinkled, for example. So when we think of genes, we're thinking of discrete transmission of properties and discrete determination of properties, that there is something in the seed, that there is something in the fertilized egg that will determine the physical properties of the organism as it matures, and that there is something in the seed that will, in the adult organism, that will be passed on to the next generation of seeds that contains the determinant of that property. So that is effectively what Mendelism proposes, and because Mendel published this in a fairly rather obscure journal in 1865. That's not the whole story, because Mendel was corresponded with some of the leading biologists in Europe, and they knew about his work, so that, in fact, they sort of dismissed it as not significant in the 1860s and 1870s. But as I mentioned in the first lecture in this series on evolution, on the idea of evolution, in the late 1880s and throughout the 1890s, a number of biologists began to recognize that evolution requires a discrete theory of inheritance. And they were looking for a discrete theory of inheritance, did what any graduate student today would do, and that is a literature search, and various people, at least 4 that we know of, 4 whose subsequent careers called attention to them, but there may have been hundreds for all we know, rediscovered Mendel's paper. And from 4 different countries, Correns, Chermak, De Vries, and Bateson. So from Austria, Germany, Holland, and England. Bateson, who coined the term genetics, who coined the term gene, I mentioned in 1905, to refer to this discrete determinant and transmitter of physical characteristics. Bateson happened to read Mendel's paper while he was traveling to give a paper at a conference in 1900 on crossbreeding. De Vries had discovered it earlier because he had developed, and in the late 1880s published, a major textbook on mutation theory. And he was the great champion of mutations as the key to evolution, not natural selection acting on continuous small variations, but mutations, which essentially replace natural selection. And so in 1900, it is a fact that the 20th century begins with a core of biologists defending a Mendelian theory of inheritance, which we have come to call genetics. Now, the gene represents something like an atom. It is analogous in biology to the atom in chemistry and the atom in physics. Let's just hold that thought for a moment, because this theory of inheritance, this Mendelian theory of inheritance, did not please everybody. And in particular, it did not please Carl Pearson at the University of London and his collaborator, Weldon. They became the nucleus, and the journal that they controlled became the center for what they considered loyal Darwinian evolutionary theory, which insisted on continuous small variations that then would spread by natural selection and create new populations with new kinds of characteristics. And they rejected Mendelian discrete theory of inheritance, and they rejected mutation theory with the jumps that they represented, and of course the downplaying of natural selection. Unfortunately, there was tremendous personal bitterness between Bateson and Pearson, and this, for a decade, prevented these communities from cooperatively working and recognizing what eventually became clear between 1910 and 1920, that there was no real conflict here. That, as a matter of fact, each needed what the other was doing. But this is, too, a fact about science. And not for the first time have we seen this kind of a phenomenon. The opposition to continental drift, you may recall, in the United States went on for over 50 years. The strongest possible opposition to any kind of geological research that was explicitly based on continental drift, but then all of a sudden in the 1960s, it emerges, it has the legitimacy of plate tectonics. Of course, there's new research, but an argument can be made and has been made that the data that was necessary to convince the Americans was there. They just chose not to read that data in a way that supported the idea of what we have come to call plate tectonics and what Alfred Wegener called continental drift. So this idea that personal rivalries can get in the way of the ideal of scientific research and progress. Science is not this disinterested pursuit of truth only. There's an interest as well. And for a decade, the Pearsonians, who promoted themselves as the loyal Darwinians, and the Batesonians, De Vriesians, who were perceived, were cast in the role of the anti-Darwinians, and it must be said that they did to some extent embrace that goal. But in fact, in 1909 and 1910, a critical change took place, and it had to do with the developments in sort of classic biology using much improved microscopes and paying attention to cellular division. By doing so, people had begun to recognize that in cell division, a crucial role is played by tiny little objects that we call chromosomes. That chromosomes, which are in the nucleus of a cell, play a critical role in reproduction and it was thought in the transmission of characteristics. In 1909, observing chromosomes in the course of cell division revealed that what we call now genetic recombination, when the chromosomes separate and recombine in all forms of sexual reproduction, whether it's bacterial sexual reproduction or mammalian sexual reproduction, when in the course of cell division the chromosomes divide and then recombine, there are all kinds of opportunities, and in fact, there are many occasions when bits and pieces of the chromosomes break off and reattach themselves in different places, or switch from one chromosome, one of the chromosomes in the pair to the other chromosome in the pair. And so this is called recombination. You can get new kinds of characteristics out of this recombination. Thinking along these lines, in the context of an aggressive debate over Mendelian genetics, naturally suggests that somehow what we mean by genes are on the chromosomes. And in 1910, T.H. Morgan, in his laboratory at Columbia University, later he moved to Caltech, Morgan founded what was probably the single most fruitful research program in biology in the first half of the 20th century, and that was fruit fly research. So it was a fruitful line of research, and it was the study of fruit flies because many of their chromosomes are extremely large from the point of view of biologists. and therefore readily observable under the microscope. And what Morgan discovered in 1910, until that time, Morgan had not been a supporter of Mendelism. He was not a genetic theory of inheritance person. But what he discovered was that he could exactly correlate eye color in fruit flies with a particular chromosome. And all of a sudden, he became a true believer. And he announced that genes are real and that they are located on the chromosomes. Of course, he was not the only one who said this, otherwise he might not have had the impact that he had. But the study of fruit fly chromosomes, the study of genetic variation, the study of mutations, using the fruit fly as the experimental subject, became a major source of research for the next 40 years in biology. And a tremendous number of careers in biology and in genetics were based on fruit fly research, which is why Caltech wanted to hire Morgan away from Columbia. So in 1910, we now have a strong scientific argument that genes are real. And from Morgan's point of view, genes really are like atoms, because here we can correlate eye color with what seems like a localized physical cause. It's interesting, therefore, to note that treating the gene as a kind of inheritance atom and taking it that it's real, it's not just a mental construct or a hypothesis, was happening at about the same time that physicists were coming to the conclusion that chemists had come to 50 years earlier, that atoms were real. Because you may remember from earlier lectures in this course that it was only in the late 1908, 1909, 1910 that the majority of physicists accepted that the chemical atom was a physical reality. Until then, they thought that it was a useful device for describing chemical reactions, but that it was not physically real. And so at about the same time that physical scientists are accepting the reality of physical atoms, biologists are beginning to accept the reality of inheritance atoms. That's one interesting affinity. Whether there's any connection or not is not clear. There's another one, however, which is, I think, much more fertile and fundamental. The Pearsonians, in pursuit of defending Darwinism at the level of the way that continuous small variations could accumulate in a population and create new species, developed mathematical models, which had not been done before. Mendel had very primitive mathematics. That's why you can teach it in high school, because you really only need to know arithmetic. And maybe a little bit of algebra. In the case of the Pearsonians, they developed progressively more sophisticated mathematical models for describing genetic patterns over generations, how genetic patterns formed over generations. And of course, the pattern that we're looking for is how you can, by accumulating small variations, wind up with a population down here that's clearly different from a population over here. In 1909, 1908, 1909, two men, quite independently of one another, a mathematician, one of the greatest of the first half of the 20th century, G.H. Hardy, and a German physician who was sort of an amateur, so to speak, in this field, Wilhelm Weinberg, published what has been known ever since as the Hardy-Weinberg law, which is an algebraic expression which allows you to see how the introduction of a variation, a genetic an heritable variation in a population, will spread and stabilize in the population. So it will reach equilibrium, except that natural selection doesn't allow equilibrium to be reached. So that natural selection will continually destabilize the equilibrium that would be reached, and the Hardy-Weinberg law gives us the first tool to understand how a mutation can spread through the population. Now, of course, mutations are not Pearsonian; they're Batesonian, and so the fact that we now have a law which can really help us understand how variations can spread if we look at the variations as mutations. But they could be small mutations. That's the difference. Is one of the insights that led to the overlapping of the Pearsonians and the Batesonians, so that by 1910 the conflict was in effect over, And now what we're looking at is how we can use insights from both of these parties in order to get a richer understanding of the relationship between inheritance theory, between genetics and evolution, the way that mutations, the way that spontaneous variations can spread in a population. In 1915, the mathematician H.T.J. Norton published tables which extended the Hardy-Weinberg law and actually showed that, let's suppose that a genetic variation gave you a 0.1% survival advantage over your siblings and peers. How long would it take for that variation to become normal in a population? How many generations would it take before everybody had it? And Norton's tables would also allow you to see how quickly a gene would be removed if it had a negative survival advantage. These tables were published first in 1915. By the end of the 19-teens, had convinced just about everybody that genetics and Darwinian evolution were hand in glove. That this was Now, what's interesting about this is that something similar was happening in physics, because these mathematical models are based on no understanding whatsoever of the underlying biological process. Nobody knows how genes work. These mathematical models are purely empirical descriptive. They have a statistical empirical character. They're not based, as, for example, most of 19th century mathematical physics was based on an attempt to model, using mathematics, what was believed to be a physical process, because we want to understand the process. The geneticists were saying, well, you know, there are people who are studying the process, and that took quite a while to straighten out, the 1950s really, but we can look at the consequences of the process and we can model that mathematically. Now, that should remind you of quantum theory in the 1920s, where Heisenberg, for example, developed his version of quantum mechanics, ignores the underlying processes. We're not interested in what the underlying processes. Even Bohr's work between 1912 and 1920 of the quantized orbital electrons and how that theory could explain spectroscopic data, that there was no attempt to really understand, well, what's really going on there? These mathematical models work. And so we have, I think, an interesting intellectual climate. I don't believe there was any influence whatsoever at the time between quantum physics, which was a radical theory in physics in the 19-teens, brand new and very radical, and what was going on in biology. Mathematics was still, compared to physics, very, very weakly employed in biology. And yet we see, for whatever reason, we see analogous kinds of conceptual responses. So between 1910 and 1920, we have a growing conviction that genes are real. What really nailed it down for the overwhelming majority of biologists. And it's not clear that it should have done it for them, but I think in science generally, it often is the case that when you look back at a theory that has been established as dominant, there is some moment when you can say, this was when everybody got on board. But if you actually say, well, why did they get on board then? Was that such a compelling evidence? Well, it wasn't logically compelling, but apparently at a certain point, people felt that they wanted to get on board. This is obviously a very subjective interpretation, and they used that as an excuse. But in 1927, Hermann Müller subjected fruit flies to X-rays and was able to induce mutations artificially. Was able to create mutant fruit flies, and then by looking at their chromosomes was able to convince just about everybody that genes were indeed real, and that they were physically located on the chromosomes. I had indicated earlier that the next step in the development of genetic theory was the extension in the 1920s of these genetic patterns to populations, studying populations rather than studying individual variation. And here, too, we see a phenomenon that we have seen before in the history of physics. In the 1920s, three men working independently and working on three different grounds. That is to say, they did not share almost any fundamental commitments in biology. Their conceptualization of the genetic process was fundamentally different, and what they were interested in modeling was different. And yet the three men, R.S. Fisher, Sewell Wright and J.B.S. Haldane came up with functionally equivalent mathematical models of the spread of genes in populations, the spread of genetic mutations in populations. This work was all published in the late 1920s and early 1930s. By 1930, 1931, all of them had published major textbooks, very influential, using very sophisticated mathematical models, by the way. And this, too, is like what we saw in quantum mechanics in the mid-1920s, when Heisenberg and Schrödinger in 1925 both formulated versions of quantum mechanics based on fundamentally different physical principles that turned out to be functionally equivalent and intertranslatable. Which was quite a shock at the time to Schrödinger, who thought his theory was much prettier and closer to the truth. And so by 1930, we've got a really viable population-based genetic theory that understands that a source of variation of biological variation is recombination, that the sexual reproduction process itself contains the key to understanding where variation comes from. So genes are real. Now the question is, what are genes? We have to step back now. The 20th century inherited a prejudice from late 19th century biology, which was that the key to life was protoplasm. It was the stuff that's in the cell, not particularly the nucleus, and in fact, the extranuclear material in the cell, that is the key to life and the key to understanding life. And in the 1890s and very early 1900s, in particular, chemists and biologists had discovered enzymes. And it was thought that enzymes are the key to life, that studying enzymes will give us the key to life. In 1907, the German chemist Emil Fischer made a major contribution to biology by showing that proteins are built out of amino acids. Amino acids are a particular kind of molecule. They come in a number of different types. There are 20 different amino acids that are that make up every single protein in the human body. There are over 10,000 different proteins in the human body, and they're all made out of building blocks like Legos of amino acids. And in 1907, Fischer showed that that's what proteins were made of, that proteins were structured. They were long molecules built up out of amino acid 1, 3, 7, 11, then maybe 15, 13s, whatever. And that he was able to synthesize them. Anyone could make amino acids, and then you could put them together. And you could build synthetic proteins that some of which actually did exist in the human body, and others did not, because those 20 amino acids can be combined in millions of ways. It turns out that we only have 10,000 of those millions actually at work in our body. He was not sure that proteins and enzymes were identical. In fact, we know that enzymes are a specific class of proteins. They are proteins that act as catalysts in reaction. They don't get changed when they engage in precipitate a reaction, whereas most proteins are changed by the reaction. Hormones are another class of proteins. So proteins, as far as Fischer was concerned, proteins is where the action is. Understanding proteins is the key to understanding life. Now, when we talked about chemistry, quite a while back, the history of chemistry in the 20th century, one of the important developments in the history of chemistry was the argument in the 1920s of whether large molecules had fixed structures. And John Staudinger was the chemist who fought the battle that ultimately was vindicated in part through the invention of the ultracentrifuge by Theo Svedberg in the late 1920s, which proved that macromolecules, that molecules could be very, very long and had very specific structures. Proteins are exactly that kind of structure. And the next step in biochemistry and genetics from 1925 to 1950 was trying to discover the structure of complex molecules. Now that it came to be believed, it's only believed in the 1920s that large molecules had a precise structure, as opposed to sort of a fortuitous combination of things, that they actually had a very precise structure. But proteins work in the body of the cell, not the nucleus. genes were thought to be associated with chromosomes which are in the nucleus. So it's not clear, we're talking 1920s, 1930s, 1940s, what the relationship is between studying proteins and genetics, especially because as early as the 1860s, a German biologist by the name of Friedrich Miescher had claimed that there was a nucleic acid, that there was a molecule that is, chemically speaking, an acid, in the nucleus of a cell that seemed to him to play an important role in reproduction and inheritance. He said this already in the 1860s. Development was relatively slow, so to speak, except for the identification of this nucleic acid as DNA. In the 1930s, DNA was studied and dismissed as relatively uninteresting because it seems to be just a mix of 4 different types of bases, and it doesn't go anywhere in particular, so it was set aside. And it was set aside in favor of arguing that genes make enzymes. One gene makes one enzyme, then the enzyme does the work in the cell. This was a theory developed by George Beadle and Edward Tatum in the early 1940s. Beadle was particularly proud that this definitively linked biology and biochemistry. It's called the one gene, one enzyme theory, and it was very productive as a research program, but it turns out to be wrong. In 1944, Oswald Avery and his collaborators were able to prove without any doubt that DNA, well, I shouldn't say that because some people doubted it for another 10 years, that Oswald Avery was able to establish for most biologists that DNA in the nucleus of the cell was the carrier of inheritance. So proteins are out as the key to inheritance and the key to life. What's in is DNA. Now, in between 1925 and the late 1940s, after Avery's experiment was publicized, there were a number of technical developments in biology and in chemistry that became critical to understand why we were all of a sudden able in 1953 to discover the structure of DNA. And these are another example of how what we know reflects how we know. And we've seen this happen in science after science. The instruments available to us are a profound influence on the kinds of theories that we formulate. In the 1920s, I told you, we had for the first time a consensus that large molecules had a precise structure, a precise chemical structure and a precise spatial structure. That the atoms in the molecule were arranged in very fixed ways. X-ray crystallography as a tool for studying structure became available in the 1920s as physicists were using them to study the structure of crystals and, in fact, various obscure quantum phenomena. But it's not easy to crystallize biological molecules. Between 1925, 1926, and 1950, biologists learned how to crystallize biological molecules without warping them. Then they could start studying their structure using X-ray crystallography. And in fact, this is what happened. In 1951, Linus Pauling used this technique, crystallizing proteins and studying them using X-ray crystallography to announce that all proteins had the same structure, which he called an alpha helix, a single helix, not the double helix. Between 1951, when he announced this, and 1953, DNA was crystallized. In 1953, Pauling in his laboratory at Caltech and Watson and Crick in their laboratory in Cambridge used X-ray crystallography to study crystallized DNA. And of course, because Pauling stumbled, Watson and Crick beat him to the line and announced that they had discovered the double helical structure of DNA. 4 years later, Matthew Meselson and Frank Stahl established that DNA reproduces by the two helices separating and each creating a complementary helix because of the way that the base pairs are structured in the peeled apart DNA. And that essentially, by 1957, essentially sealed the course of genetic theory since 1957. That is the model, that genes are carried by the DNA. So what happened to the protein? Well, what do genes do? Genes on the DNA make proteins. And we'll talk about that in the next lecture.